Okay, well, this is chapter 8 on enzymes. We will be focusing not only on enzymes, but a little bit about metabolism. We'll talk a little bit about free energy and some basic principles about energy. Now, we'll revisit that when we get to Unit 3 on cellular respiration and photosynthesis, because those are two metabolic reactions that we really talk about in AV biology, is cellular respiration and photosynthesis. Our main focus here is enzymes. So you can't really understand how enzymes change reactions unless you understand what a reaction is. And so that's why we start with kind of background information on metabolism. This is actually sections point 1, point 2, and point 3. There's some other information in there that we revisit when we get to the next unit. Okay, so metabolism is the totality, the total of an organism's chemical reactions. So you probably heard before metabolism. You have a sluggish metabolism, or you have a speedy metabolism, or take this and you can increase your metabolism. They're talking about how you break down food, and that's usually what we're talking about. We talk about that metabolism, but metabolism in general is just all the chemical reactions that happen on the cellular level in your body, or in any living thing. Each step of a pathway is catalyzed. That just means that it is made to happen by a specific enzyme. This is important here that it's specific. Specific enzymes work on specific reactions. Each enzyme works on a reaction, maybe one or two reactions, but enzymes are very specific. So therefore, you have thousands of enzymes in any given cell. Catabolic reactions are reactions that release energy by breaking down complex molecules. So maybe you're breaking down protein or a carbohydrate into simple sugars. Anabolic reactions consume energy. So you have to put energy into the system. And remember, the cellular energy is ATP. And the reason you need energy is because you're building complicated molecules from those monomers. And so you need energy to build that. And you think about it this way, it's always going to take more energy to build something than it is to just tear it down. So here's just showing you that maybe this is your starting molecule. Maybe this is some kind of a carbohydrate. And you're going to break it down into simple sugars. So the starting molecule is going to turn into another molecule, and that's going to be catalyzed by one enzyme. And then that molecule is going to be broken down into another one, and that's going to be catabolized by another enzyme, and then that one's going to be broken down in the final product. Now, this is only a three-step process. Most chemical reactions that happen in your body are going to be more than a three-step. Take, for example, cellular respiration, which we'll talk about in our next unit. Cellular respiration, we're going to actually model some parts of that chemical reaction, and you'll see that it's many, many steps. Matter of fact, we'll only be able to model a few steps because of the number of students we have in the class. But that can be summed up in many, many steps. I'm not sure exactly how many steps, but there are a lot. And each time one molecule is turning into another, you are going to have an enzyme that takes care of that reaction. So cellular respiration, what you're actually doing is you're taking sugar that you eat, that you've already broken down from the carbohydrates, plus oxygen, and you're going to release water, carbon dioxide, and also form energy. So this is the reaction, and this doesn't happen in 1, 2, 3 steps. Like this reaction, this is going to happen on a lot of reactions. Glucose is not going to turn automatically into ATP. It's first going to turn into something else, and then something else, and then something else, and so on and so forth. And so there's a lot of transformations that happen in any chemical reaction. Energy is the capacity to cause change, the ability to rearrange matter. You may have heard of this before, is the ability to work. Okay. Chemical energy is the potential energy available for release in a chemical reaction. Now, it's probably a different answer if you ask a chemistry teacher or a physics teacher or something, but in biology, That's going to be our definition. Organisms are energy transformers. That's what they do. They transform energy. The thermodynamics. There's a couple of really important laws that you just need to kind of understand. If I can get to that slide. Oh, there it is. Um, the first one is that energy cannot be created or destroyed. It has to be transferred or transformed. This is also kind of like not only energy, but atoms. So, for example, if we have water and we're going to turn that into or break it apart into water and oxygen, that's supposed to be a plus sign. I'm not doing real big chemistry here. We know that if we have two hydrogens here, we have to have two hydrogens here. Well, we've got two oxygens over here. Well, we only have one here. So we're going to have to put two here. Well, that makes that four. So that means we have to put two here. And so you have to balance it because nothing can actually be created or destroyed, atoms or energy. Most energy transformations convert a good portion of usable energy to heat. Think about body heat. So this is how we get body heat. It's because all the chemical reactions that are occurring in our bodies are going to produce body heat as a byproduct. My husband's always hot. And we always joke because when he was younger, he had a really high metabolism. And one of the products of that is just being hot all the time. He's not as hot now that he's a little older, but he also cannot eat an entire bag of potato chips anymore. Entropy is the measure of disorder or randomness in a system. Now, I'm not sure I'm ever going to fully understand entropy because it doesn't make sense to my brain. That's probably more of a physics thing. And if you ask Mrs. Brockman and Mrs. Hobbs, they could probably explain entropy to you a little bit better than I can explain it. Also, you will have a Bozeman video over free energy enzymes. And I think Bozeman does a pretty good job of explaining free energy enzymes. Me, not so much. The second law of thermodynamics is that every energy transformation increases the entropy of the universe. So how does an energy transformation increase entropy, which is disorder in the universe? And that's one of those things that the book tries to explain it. I'm not sure I will ever get it. If you're really philosophical and enjoy physics, you might want to read that part of the textbook in chapter 8. Myself, I just kind of take it as fact and accept it and assume that the physics people that have come up with these are a lot smarter than me. Needless to show the two laws. So the first law is that energy cannot be created or destroyed. So here you're just transferring the chemical energy into the bear. The bear is then going to do chemical reactions through the processes of cellular respiration. And the bear is going to produce carbon dioxide and water. Those are products of cellular respiration. And also, notice he's going to produce some heat. And that is a result, remember, of transforming energy. This is probably more important about thermodynamics for us anyway, is that spontaneous processes occur without energy input and increased entropy. Again, this part is like one of those, okay, I accept it, but I don't really get it. They occur without energy input, okay? So this is without ATP. Entropy of a particular system, such as an organism, may actually decrease. So this gets more making sense to me, that you can actually decrease entropy, which is disorder, so another increased order, because think about cells, they're very orderly, as long as the total entropy of the entire universe increases. Well, that's very philosophical, very physics, very not my field, but that's where you can kind of get around the idea that, yes, cells are very organized, but we're looking at the entropy of the entire universe, not just the cell. So free energy used to be called available energy. It's probably actually a better name for it. It's just the energy that's available for use. The portion of a system's energy that can perform work when temperature and pressure are uniform throughout the system. So, for example, a living cell. This is the equation for calculating free energy. You'll also hear it called Gibbs free energy. I'm sure that has something to do with the original scientist that came up with the equation, but he's not important for what we need to know in AP biology. Delta H is the total energy in the biological systems. T is the absolute temperature in Kelvin. I'm sure you're familiar with that from chemistry. And delta S is the change in the system's entropy. So really, why is this important? What can we get from this equation? Am I ever going to have you calculate this equation? No. Is the equation on your AP formula sheet? Yes. I have never seen it used on an AP exam. But because it is on the formula sheet, technically it's their game. They could ask you a question over free energy, thinking they're probably never going to do that. But all of this information here is available, and it's a simple mathematical equation. What is important to understand about this is that a chemical equation is spontaneous when delta G is negative. That's what's important to take away from this. So if you were to have an equation and the delta G is negative, that reaction is going to occur spontaneously, which means that it is going to occur without ATP. Okay, that's what's important to take away from this. So exergonic reactions are spontaneous reactions. Okay, these have a negative delta G. They have a net release of free energy. So where does this play in for us? Cellular respiration is an example of an exergonic reaction. It does not take or consume ATP as a net. It does use some ATP to make happen, but the net is a production of ATP, not a use of it. Endergonic reactions absorb free energy. How do you remember these? Ender, into, indo, end, kind of like the in. It's a root word. Exer, exit, kind of thing. They are going to release free energy. They absorb free energy from its surroundings. Okay, so they're going to use ATP. And they store free energy in molecules. The delta G here is positive. So an example of this kind of reaction we'll talk about is photosynthesis. So photosynthesis actually uses ATP. Where does it get the ATP? In the cell from cellular respiration, where we produce it in the mitochondria. So it's going to use ATP, and then it's going to store energy in its leaves. How does it store the energy in its leaves? Well, in this case, it's going to store energy as glucose or as sugars. And probably not simple sugars like simple glucose, but probably going to store it as more complex carbohydrates like cellulose. Here is showing exergonic and endergonic reactions kind of in a graph way, a visual way. I like visual ways, so this makes a little bit more sense to me. So in an exergonic reaction, you have energy released. Okay, this is a spontaneous reaction, and the delta G is going to be negative. So here are your reactions. You're going to release energy, and then you're going to form the product. So it's kind of going on a downhill slope. And then over here, endergonic are going to require energy, and they're going to be non-spontaneous. So they have to actually have something starting that reaction. So the reactants are going to build into the products. The delta G is going to be positive, and you're going to need energy being put into the system. In this case, in photosynthesis, we start with the sun's energy. That's the main source. And then we also involve some ATP. Enzymes are molecules that act as catalysts. Now, it's a macromolecule. Do you remember which macromolecule? If you said protein, you are correct. That acts as a catalyst. A catalyst is a chemical agent that speeds up a chemical reaction or a reaction without being consumed. That's important. Enzymes go in, they make the reaction happen, but they come out unharmed. They look exactly like the enzyme that went in. You can reuse enzymes multiple times as long as you have enough reactant to keep the reaction going. Activation energy is the energy required to start the reaction. Often that's supplied by thermal energy or heat. And I usually, in class, when I used to do sometimes strike a match is like this great show of activation energy. It provides a barrier that determines the rate of the reaction. In most reactions, that activation energy is so high, the reaction will not proceed at all. Enzymes work by lowering the activation energy. That's really important. This is their job, okay? When you ask, how do enzymes speed up reactions? Because that's what they do, okay? They're catalysts, remember? They're biological catalysts. And they speed up chemical reactions. Well, how do they do it? They lower the activation energy. They make sure that what's needed to make that reaction happen is low enough to actually be obtainable. So here's an example of a graph, and you should understand this graph and what it means. This is the progress of the reaction, so is it happening or not? And this is the amount of free energy. So we start with our reactants, and we go up to this transition state where we're kind of like we could make this happen or we could not make this happen. And then if the reaction actually hits that activation energy, the reaction will actually form the products. Okay? Now, in this case, this is a reaction where our delta G is negative, and so this would be a spontaneous reaction. Remember those graphs before I showed the spontaneous and the bar graphs kind of going down. And then here it's showing the course of a reaction without the enzyme. So without the enzyme, notice that this energy that's required is way up here. With the enzyme, the energy that's required to be put into the system is down here. So there's a difference between that amount of energy that is needed to make that reaction happen. So what we've done, or the result of this, is that the activation energy, that's what this means, the activation energy with the enzyme is lower than without it. And then the free energy is unaffected by the enzyme in this case. Enzymes are specific for the reactions that they catalyze. Remember, I talked about that before. And remember, we go back to our first couple of chapters where we said that shape is important for function. Okay? The shape of any molecule is important for what it does. In this case, the shape of the enzyme is specific to the substrate that it works on. Okay? That's why, really, we have only one enzyme working on one or two reactions. Many enzymes have specific locations within the cell as well, and some can move around inside the cell. So some are like maybe just within the mitochondria, and others can actually travel throughout the cell. The substrate is the reactant that the enzyme acts on. Okay, so we're basically going from substrate to product. So it's another way of calling another word for the reactant. The active site is the restricted region of the enzyme molecule that usually binds to the substrate. This is important here. It binds to the substrate. And remember, the shape is really important. Okay, the shape of the active site fits perfectly with the substrate. I always like to think of this as, and this is often referred to as, the lock and key model. I remember that from Biology 1. You cannot take your house key and unlock my house door because your key is specific to your locks. You cannot take my car key and start your car because each lock is specific to the key, or each key is specific to the lock that it is working on. And it's the same basic idea. The active site is the area where the key binds to the lock. And in enzyme, the active site is the area where the substrate binds to the enzyme. It's actually not, they used to think it was a very rigid area, but it may change slightly as the substrate binds. We call that now an induced fit. So when the substrate binds, it may actually change shape a little bit at that binding time. So here is an example of an enzyme. So this is made up of lots of little atoms. Okay. Remember that an enzyme is a protein, and proteins are made up of amino acids. Okay. Remember, proteins have a primary structure, a secondary structure, a tertiary structure, and possibly a fourth structure. The active site is right here. That's where the enzyme is, or the substrate is actually going to bind. So say this is water, and we're going to break water down into hydrogen and oxygen. And remember, we've kind of established before that we have to balance our equations. So we're going to break down the water. So the enzyme is going to come in, and it's actually going to hit that substrate there, and then it's going to break it down. This is probably a really bad example, because it's not really a biologically related one. So here's kind of the scheme of things, step by step. The substrate is going to enter the active site. Okay, they're going to be held there by a interaction, not really a bond, but a weak attraction. The active site can lower the activation energy, and that will speed up the reaction. Remember, if we don't have the enzyme here, which is what we want, then we might not have that happen. The activation energy might just be too high not to reach it. The substrates are then converted to the products, and the products are released. Remember what I said before, the enzyme doesn't change. Okay, the enzyme remains there. So you can reuse this enzyme multiple times in any given cell. And it's now available to pick up new substrates and then continue the same process. As long as you're continuing to supply the substrate, the enzyme should be fine. It shouldn't run out because it's always there. Now, if you reduce the substrate, then eventually you're going to turn all the substrate into product and you're done. Let's see. So just as you can take two substrates and turn into two products, you can also take two substrates and turn them into one product, or you could take one substrate and turn it into multiple products. That's where we get into, are we building things or are we breaking it down? It is a catabolic reaction or an anabolic reaction. The entire cycle happens quickly. A single enzyme can act on about 1,000 substrate molecules per second. So one. Inside your body right now, a single enzyme works on 1,000 substrates at once. That's why we can make reactions happen, and we can do it very quickly. Most reactions are reversible, and the enzyme that catalyzes their forward and also their reverse reactions. So they can build and break down. So there are some things that will actually affect enzymes. Enzymes have an optimal pH range that is not necessarily set. For example, the enzymes in your stomach are probably going to operate at a lower pH level than the enzymes in your blood. So remember, your blood is about a pH 7.4. I'm not sure exactly what your stomach is, but I want to say maybe it's like in the 6s or 5s. No, wait, maybe it's in the 4s. I looked that up at one time, and I just can't remember off the top of my head. But the enzymes that are in your blood are going to operate at a different pH than the enzymes that are in your stomach. That's the whole point of that. Also, enzymes have an optimal temperature that they can operate at. So, for example, in the human body, we are about 98.6 degrees Fahrenheit. That's our temperature. And so all of the enzymes that are inside the body are going to operate about that temperature. Now, if you start running a fever of 106 degrees Fahrenheit, wow, you're getting into brain damage. And the reason that you're getting there and the reason that you could die from a fever that high is because the enzymes that are in your body or in your cells are not going to operate. All of those chemical reactions, your metabolism, as we call it, all those chemical reactions are going to stop or they're going to slow down because the temperature is outside of its optimal range. Now, our lab that we're doing, the AP Enzyme Lab, is going to address some of these issues with enzymes and what they're looking at, pH and temperature, things like that, and how that affects the rate of a chemical reaction. Anything outside the specific range may cause the enzyme to denature. Remember, we talked about denaturing in another chapter, and denaturing just basically means to change or to break down the protein. It takes that protein structure and it breaks it up. Remember, it takes that primary structure and destroys it, or the secondary structure and destroys it, so that it's no longer able to function. So this shows just a couple of enzymes and where they're optimally active at. So, for example, the chemical reaction might still happen at a different, not the exact optimal temperature, but you'll see that it's like a bell curve. On this end, it's not exactly at its best. Here, it's absolutely not going to work, but there is an optimal temperature. So this is an enzyme in the human body. This is about that 98.6 degrees. Now, however, if we're looking at a bacteria that's very heat tolerant, their optimal pH or optimal temperature is actually here, closer to 80 degrees Celsius. When that bacteria gets down to where we're optimal at, 37 degrees Celsius, it actually would not be active. Because right around 40, it's not even active. So remember, just because all enzymes have an optimal temperature does not mean that every enzyme has the same temperature that is optimal. And then here's the same thing with pH. So this is an enzyme in the stomach, and this is one in the intestine. Your intestine is far more basic than your stomach. And so, I guess the stomach is right around there. I didn't think it was that bad. So the optimal pH for the enzyme in the stomach is going to be around 2-ish, whereas the optimal pH for trypsin, which is in the intestine, is going to be around an 8-ish. So if you put that trypsin enzyme into the stomach, it's not going to survive. It's not alive, so it can't really not survive. It's not going to work. It's going to be denatured. Not work. If it's outside the range, it can be denatured. Cofactors and coenzymes. Cofactors are non-protein parts that bind to the enzyme. Okay? Coenzymes are cofactors that are organic. Many vitamins are coenzymes, like coenzyme A. I know that's an example of one. These basically help. They bind to the enzyme and they make the enzyme work better. These are helpers to enzymes. We're going to stop right now on this, and then we're going to come back to the rest of this in another video. Oops. And talk about activators of enzymes.